So it is the 17th of June, and this will be the first lecture related to exam number 3. Exam number 2 is available for you to review. The grades were posted yesterday. It was not as pretty as the first exam, but you're aware of that. And I kind of warned you that the second exam is a little bit of a wake-up call for many students. As I look back over the YouTube, I can watch the number of hits on my YouTube sites, and I saw that the numbers dropped a little bit. And that's okay, but I've had students ask me, what is the one thing that I can do that will likely improve my grade? And my answer is there's only so many things we can do, right? There's only so many hours in the day. But if there's one thing that you might do to give you greater success, might be to review the lectures a second time. They're on there. They're on YouTube for you. You can easily pull them on your smartphone or your tablet or your home computer. And that might be the one thing you could do. That'd be an extra 4 hours or so a week, but it might be the best time you spent. And while you're listening to it the second time, a lot of ideas will synthesize and will come together in ways that perhaps they didn't the first time through. Now, if you don't want to do the reviewing of the lectures, you might consider joining a group. Right? Obviously, joining a study group is a great way to approach this material as well. I don't want you to get frustrated, though. I don't want you to get really discouraged by the second exam scores. They tend to be a little bit lower. As I said to you before, the endocrine system has a lot of details, and we'll be diving into each of those hormones, into each of those systems as we move through the rest of the course. Now, the exam is available for you upstairs. You can review it. Hillary will be there again tomorrow afternoon. So she's there on Monday and Wednesday afternoons and Tuesday, Thursday, and Friday mornings. She has the key. She has your exam booklet. I always encourage you to stop by and take a look and see where you could have done better and learn from those things that didn't go well, and also confirm the things that you do know well. The next exam, chapters 11, which is what we're discussing today. This is muscle. We'll be spending most of our time discussing skeletal muscle, but toward the end we'll also pick up on cardiac and smooth muscle. We'll learn more than you ever thought was possible about muscle. Then we'll move into some blood, and that's where we're starting tonight in lab, and on Thursday in lab we're doing a hematology lab, and there'll also be some cardiovascular on this next exam. So this next exam is largely muscle, right, of your body as well as of the heart, cardiac muscle, cardiovascular system, and blood. That exam is when? What do you have on your syllabus? July 1st. So that means you get to go and enjoy July 4th free, right? You won't have to do a lot of studying that weekend. So we'll have that next exam on July 1st. Remember that there are guided reading assignments to help you with this material. There are the Connect assignments for you to do online. There will be quizzes online as well. Keep in mind, too, as you're doing the quizzes, there are going to be this semester 14 of those online quizzes through Connect. Only the top 10 scores will count. So if you miss a couple or don't do well on a couple of quizzes, know at the end of the semester, 4 of your lowest grades will be removed from your overall grade. There will also be, at the end, 2 extra credit quizzes. They're in the syllabus, there's a mention of it, but those will be true, completely extra credit opportunities for you later in the course. So my word to you is just hang on. And as we move through the course, there will be some grade-raising opportunities that will come your way. And I want to encourage you. If you have any questions at all, though, please let me know what concerns you have as we go into this next exam. Now, Chapter 11, right? We're dealing with muscle here and muscular tissue. There's a few slides I'm asking you to review on your own. They're largely concepts that you should be carrying over quite comfortably from Biology 105. There's a little red asterisk on the slides. I'll touch on them very quickly, but I'm not going to spend much time on them at all. Let me stop right there. Looking at your syllabus, any concerns, any questions, any housekeeping issues?
The case study.
The case studies. The case studies will be due next Thursday, and I'll accept them through lecture time.
So the peer review is the same?
So for some of you who had lab today or tonight, your lab reports are due, your peer review lab reports are due next week, and then the final copy due in 2 weeks. And your case studies are due next week. So I'll accept them through Thursday lecture time, a week from, so 9 days from now. So I'll be giving you some time in lab tonight or on Thursday to get with your groups and to confirm and finalize any details related to the case studies.
So the 26th.
Right, Thursday the 26th, correct.
And then the 19th.
Then the lab report the week before that. Right. Okay. Any other housekeeping concerns? Deadlines, upcoming events? Okay. Well, so the muscular system, and tissue specifically. So you'll be reviewing slides 2 through 9 on your own. But what you're going to find there is just an introduction to what muscle is, and you know this. But I do want to point out the characteristics on this slide. If I were to ask you, what are the characteristics of connective tissue? What would you tell me? We know this one. Connective tissues, bone, cartilage, blood are all related to each other because they have what? What's the mantra? You know this. Loosely packed cells surrounded by a ground substance and protein fibers. If I were to ask you what are the characteristics of epithelial tissues, you should be able to tell me tightly packed cells, right? Avascular, always sitting superior on top of a basement membrane and connective tissue below. If I were to ask you what are the characteristics of nervous tissue, right? We've already talked about nervous tissue. You would tell me excitable, right? They can send electrical signals quickly through the body. And now I ask you, what are the characteristics of all muscle tissue? And here they are. They're excitable as well. So the moment you hear me say excitable, your mind should be going to the idea that there must be a resting membrane potential, right? Much of what we learned in the nervous system in that beginning about resting membrane potentials and about how cells have a different environment inside and outside, that same story is going to inform us all the way through this muscle unit. So muscle cells are responsive. They are excitable. They respond to stimuli. They also are conductive. That is, they can have electrical signals traveling through them. Well, where do those electrical signals come from? From the neuron, right? From the nervous system that is going to excite the muscle cell. And then we're going to see what that excitation does as it drives the muscle to, number 3, contract. Right? It's going to shorten. And as that muscle contracts, it's going to pull bones, or it's going to squeeze and push digestive products, or it's going to squeeze and empty the bladder. It's going to squeeze or relax and allow blood flow to be regulated through your body. And then it's extensible. Now that's a fancy way of saying is that it's capable of being stretched between contractions. Remember, muscles are only good at getting short. And as they get shorter, they are able to bring power and force in. But muscles also have to be, if you will, restarted, right? Brought back to their original length. So there's always this idea of extensibility. And then finally, muscles are also elastic. They have elasticity. They're able to be stretched and returned back to their original place. So we'll look at these different characteristics of muscle tissue. If I say skeletal muscle, what comes to mind? Visually, what are you going to look for under the microscope? Striations, right? So strong striping pattern under the microscope. You know a little bit about what those stripes are formed by. Remember, there were the sarcomeres and there were the overlapping myosin and actin. You have the basics of this. Now we're going to tell you how it works. So you know a lot of the parts. You know the pieces. You know a little bit about the thin and the thick filaments. But now we're going to talk about, okay, when that electrical signal comes in from the neuron and it excites the muscle cell, what are the steps sequentially, 1, 2, 3, 4, 5, that are going to be necessary for that muscle to contract? That's where we're heading. You're going to know this story about muscle contraction as well as you knew the story about action potentials in the nervous system. Very sequential. You remember that this is also voluntary muscle, right? This is muscle that is controlled by your nervous system. And recall in a vocabulary way that a muscle cell is also called a what? Muscle fiber, right? So that term fiber. Now, that term, not to be confused, what was a fiber back in nervous system? A nerve fiber is also called an axon, right? Not the whole neuron, right? So in the nervous system, a fiber is just the axon. In the muscle system, the fiber is the entire cell. Yeah, so slight different use of the term. Now remember too that these skeletal muscle cells are very, very long. They can be 30 centimeters. They can be 12 inches long. Think about your sartorius, right? That long muscle that kind of wraps around your thigh. That's a really long muscle. And the muscles within that, or the cells within that muscle, are as long as the muscle is itself. So these are very, very long cells. They're multinucleated. So they have more than one brain, if you will, right? But they kind of cooperate well together. So you notice these pictures coming from Anatomy and Physiology Revealed. Again, you can see those strong striations, and we will go back and look at the A and the I and the M and the Z bands, okay, as part of a review. We discussed it a little bit last semester. We'll come back to it. I also want to remind you that muscle doesn't exist in isolation. That muscle is protected by connective tissue layers around the outside, as well as within the layers of the muscle. So you've seen these terms before: the endomysium, the perimysium, and the epimysium. You may hear others pronounce them endomysium, perimysium. Either pronunciation appears to be acceptable. I hear them both all the time. Remember the endomysium. The endomysium, endo meaning within, so you're talking about the most delicate layer inside, wrapped around each and every muscle fiber, right, around each muscle cell. Then the perimysium, think perimeter, around. The perimysium is around what? Not the entire muscle, but instead just around each fascicle. Remember that term fascicle? Fasci means a band or a bundle. So we're talking about a bundle of muscle cells grouped together, and that would be surrounded by a layer of perimysium, and then the entire muscle around the outside of the entire belly of the muscle would have around it some epimysium. You know, epi means on or upon. So the vocab helps us out there. There's also a fair amount of collagen in these connective tissue layers, and that makes sense to you because collagen, when you hear the word collagen, what first comes to mind?
Elastic.
Not elastic, remember? I heard it. Strength, strong, right? Collagen is the steel. It's the reinforcing steel of a tissue. So when you hear about collagen, you're thinking strength. When you hear about elastin, right, then we're talking about elasticity, stretch of the fiber. So your muscles are really strong, and your muscles are attached to your bones through what structures? Tendons, right? Now we know those are very strong as well. So those are also made up of a lot of collagen. Why would we want a fair amount of collagen in our muscles? At least in the connective tissue layers around our muscles. We don't want to rip a muscle off a bone, do we? I mean, we never want to be able to pull so hard that we rupture a tendon. Those never heal, quite right. So we never want to stretch a muscle and destroy a tendon-bone connection. That can be a real problem. But also that collagen that's associated with the tendons gives the muscle extra strength. Not only can it pull, but it's also being, if you will, backed up by the power, by the strength of the collagen fibers. So again, remember a muscle fiber, aka a muscle cell, is such a uniquely different shaped cell that there are some different terms. In Chapter 3 of Biology 105, we would have talked about cells having a cell membrane and having a nucleus and having the different organelles. And that's true of a muscle cell as well, but because it's such a specialized cell, there are some different terms. Sarcolemma. New word, perhaps, for the cell membrane of a muscle cell. The prefix sarco, muscle. Right, so anything with sarco in front of it, you're thinking muscle. And lemma, remember, means rind or peel. So the rind, the layer around the outside of a muscle cell is the sarcolemma. And then rather than calling the inside of the cell the cytoplasm, we're instead going to say it's the sarcoplasm. Now, what's in that sarcoplasm? What are we going to find in huge abundance within? A lot of mitochondria, perfect, because energy is necessary for muscle. So there's going to be a lot of mitochondria. And what else is largely going to be taking up that sarcoplasmic space? That's the next bullet, myofibrils, right? And you know them better as what? You know the fibrils as the structures of which even smaller are the thin and thick filaments, right? So these proteins that are going to be contracting and sliding over each other, giving muscle its force. A couple other things in great abundance within muscle. One is glycogen. You've heard of glycogen. It's a storage molecule of sugar. So your muscles have to be ready to go at any moment, and your muscles store extra energy in the form of glycogen. It's a long polymer of glucose, sugar molecules. And then also within your sarcoplasm, you would find a lot of myoglobin. Now, I haven't discussed myoglobin before. It sounds a lot like hemoglobin. It sort of does the same job. Remember, what does hemoglobin do? Carries oxygen through your blood to the tissues, okay? But also, myoglobin sounds like a similar protein in the muscle. Okay, so your muscles have to be ready to work. They need to have extra energy, sugar, glycogen. They need to have the ability of making ATP, a lot of mitochondria, and they have to have a special oxygen-carrying molecule called myoglobin. So specialized, right? Specialized protein found in muscle so that your muscles are always ready to go. And what color is muscle? Look at any model, look under the microscope. Muscle is largely pink or red, right? And myoglobin also gives muscle tissue, like hemoglobin, a reddish tissue color. I've already said these terms. Again, these have little red asterisk on the top. I'm always fearful of jumping ahead and saying review them on your own, right? So we want to be reminded that multiple nuclei are found within muscle cells. How did these cells get to be so long? How can you have 30-centimeter long muscle cells? Developmentally, right? Many cells linked arms, if you will, end to end. Those initial cells were called myoblasts. Now you recognize that term. You've heard of osteoblasts and chondroblasts. So myoblasts must be cells that do what? Blast, create, form new muscle. And that's exactly right. So these myoblasts were individually formed, and then they linked, if you will, together into one very long cell, but the nuclei were maintained. So we have a multinucleated cell. There are also some satellite cells present in your muscles. These are basically your stem cells. Recall that your skin has stem cells. They can help to regenerate skin if there's an injury. Your muscle has a limited ability to regenerate, but your skeletal muscle has more than your cardiac muscle. When we get to cardiac muscle, we'll learn, and you know this from practical experience, that when one suffers a myocardial infarction, a heart attack, the damage is done. And it does not regenerate very well at all. So cardiac muscle doesn't have these satellite cells, doesn't have the ability of regenerating as well. But skeletal muscle, you can damage it, and you can expect to get some new muscle fibers formed through the activation of these stem cells, these satellite cells. And as you've already mentioned, there'd be a lot of mitochondria there. There's a specialized organelle called the sarcoplasmic reticulum. Sounds a lot like what? Endoplasmic reticulum, and it's not the same, okay? But it is a series of membranes, right? Remember, reticulum means a network. So it's a series of membranes, but they're found only in sarco, in muscle, and it is a series of membranes. The purpose of the sarcoplasmic reticulum is to be a calcium reservoir. Now, we discussed the importance of calcium in the nervous system, and you know that calcium was critical for action potentials and nervous conduction. You're going to be tired of calcium, right? Because calcium is also going to be critically important in muscle contraction. And we know that we get that calcium, that homeostasis of calcium, through the skeletal system. There's also going to be the terminal cisternae. I'll show you pictures of these in a moment. But at the very ends of these sarcoplasmic reticulum layers within the muscle cell, there's going to be, what's a cistern? Let's start there.
Like a well.
Like a well, right? Like a deep well, a cistern. And terminal cisternae. So they must be deep wells at the end. Okay, so the name sort of tells us what to expect. So we have the terminal cisternae, and these are little end sacs at the end, and we'll see how they interact in the cartoon in a moment. There's also T-tubules. These T-tubules are going to be these little infolds, these little invaginations that are going to be in the sarcolemma, in the membrane of the muscle cell. And we'll see how they're going to receive the electrical signal that comes from the neuron. Together, there's a triad formed, a group of three formed between the T-tubule and two of these cisternae. Let's take a look at this picture. This is a cartoon representation of a muscle cell. It doesn't look like any kind of cell you've labeled in the past. We know neurons look very different than your typical cell, so too muscle cells are very different looking cells. Let's take a look. The yellow wrapped around this cell is what? That is the plasma membrane, but we are going to call it instead the sarcolemma. Right, the sarcolemma is the rind around the outside of the entire cell. I can see, even in this cartoon, there's one nucleus, two nuclei, right? So this is a multinucleated cell. You see the purple nuclei. I can see the striations, right? If you look, you can see the evidence of overlapping fibers, overlapping fibrils within this. This entire structure that's hanging out here, that would be a what? Myofibril. And the myofibril is even further. See these little guys sticking out? Those would be some of the myofilaments. Remember this conversation? We've had this. It's been a while. So the myofilaments, you know as thick and thin, actin and myosin. They group together as a myofibril. And I said to you a moment ago that within the muscle cell, what do you see? It's stuffed full of myofibrils. And squeezed in between them, there's one, here's one, here's one, lots of mitochondria. Okay, so a lot of mitochondria, a lot of these myofibrils which will break down structurally. Now, look at this blue web. See this blue web that's spread around the cell? You tell me, what's that blue webbing? Yeah, that's that sarcoplasmic reticulum, this network of membranes that's wrapped around these myofibrils. And I told you it's a reservoir for calcium. So the cell has lots and lots of calcium at its, available to it all the time. And then look at this triad. You see the two blues and the pink? So that's the triad that I just introduced. And the two blue are the cisternae. Notice at the ends, right? So if you look at this webbing, there's these larger areas at the ends of each of those cisternae with the T-tubule, the pink structure, running down the center. And we'll see the significance of those as we move forward. Okay. So let's dive in even deeper. This should also be review, but let's dive in deeper. The myofibril, that is that larger unit. It is composed of smaller myofilaments. You know them as thick and thin, or myosin and actin. So tell me what you remember about the thick filaments. Give me 2 or 3 ideas about the thick filament. Name it. What is it doing? The thick filament is myosin, right? Myosin. It ends in IN. It's a reminder that it's a protein. It is it's a larger protein, so it's called the thick filament. It literally is larger. It looks like a little golf club all kind of matched together. I always imagine this looks like Velcro. If you've ever seen Velcro under the microscope, it sort of has these, little golf club like fingers reaching out ready to grab onto something. And I think that's all I'm going to say right now about myosin. We'll see it in repeated images. Then the thin filament is actin. Now the thin filament is not only actin. I may have described it to you that way in 105 as a simplistic idea. But the thin filament is actually more than just one protein. It's a combination of proteins. What you see in the thin filament is thinner, right? It's not as big in diameter. Each of these individual singular round proteins is a G actin. G for globular, right? A little glob, a little round protein. Those little G actins create a little chain that we now call F actin or fibrous, making a fiber, a long molecule of actin. Okay, so if you were to look at a thin filament, what you're actually looking at is a helical, a winding together of two of these F-actins. But that's not all. Even in addition to the F-actin molecules, there are two visitors. There is a molecule called tropomyosin. We'll see the significance of that tropomyosin in a moment. Tropomyosin is this long cord. It looks like this long cord that's wrapped around, okay, that light blue or gray colored. And then there's also going to be another molecule called troponin. And troponin, it looks like a little Matryoshka doll here, right? A little snowman, a little Matryoshka doll in yellow. And that molecule, troponin, is going to be the actual calcium binding protein. So as we start talking about calcium in a moment, which protein binds the calcium? It's going to be that troponin on the thin filament. Now that's not all, right? We've talked about thin and thick filaments in the past. What is new to you is that there are also elastic filaments within your muscle. There has to be the ability for it to recoil, to be pulled back to its original length. And the largest protein in the body ever described is this one, and it's called titin, right? Titin, huge, right? If you hear Titanic, right, or Titan, something really, really large. So this is a really large, huge spring-like protein, and it's going to be on each side of the thick filaments, on each side of the myosins. And we'll see that it's going to help stabilize the muscle, but it's also going to prevent the muscle from overstretching. So it's like this, those big springs on a trampoline. That's how I picture these titin molecules. Big old springs that aren't going to let the muscle overstretch. Because again, if the muscle overstretches, we could do what? We could injure the tendon, right? So we want to make sure the body has lots of mechanisms in place to minimize damage to itself. So we have the thick and the thin filament, right? On top, we see the characteristic purple in these images. We're looking at the thick filament. Then we see the thin filament down below. That's still not all. There are other proteins involved here that are regulatory proteins. We're going to call the actin and the myosin the contractile proteins. It makes sense, right? They are the two proteins that are interacting and sliding and causing the contraction of the muscle. But there are other proteins here that are the regulatory proteins. And they're going to act like switches, and that's going to be the troponin and the tropomyosin. Okay? So the troponin and the tropomyosin are going to help regulate the contraction of muscle, so we'll call them regulatory proteins. So you've seen this image, or one like it, and you know that your muscle is made up of these, thousands and thousands of overlapping thick and thin filaments. And this is just a little tiny glimpse of it. Remember the warning I gave you when we were doing the nervous system? So we're going to break this thing down, and it's going to seem like it's molasses, right? The action potentials are traveling down the axon, and then there's this, and then there's that, and then there's this. We're just showing you little tiny glimpses of these images to capture the idea, and then you're going to have to use your imagination to actually imagine this working in 3 dimensions and bring that story together. You know what I'm always saying about stories, right? The facts are nice to know, but we need to be able to tell the story of how this is working. So we see here overlapping fibers. Every time you see a picture of actin and myosin overlapping, they always make it look like it's 2-dimensional, right? It's always like my fingers, my right hand are the myosins, my left hands are the actins. And it always looks like it's just a 2-dimensional stacking of these proteins. Keep in mind, these were in those myofibrils. They are in cylindrical arrangements. And so what you have is more like, I'm forgetting the name. What are those plugs, right? With the round end to them, with all those little connections, right? So it's almost like you've got overlapping actins and myosins, and they're in a cylindrical arrangement. They're in overlapping arrangements. So very densely overpacked. Not this simple stacking 2 dimensions, right? 3-dimension overlapping structures. And what we'll see is that in this section of the muscle, and you know this term, sarcomere. What does sarcomere mean to you by just a word? Sarco? Muscle. Mere? A polymer is a molecule made up of many parts. A monomer is a molecule made up of just one part. So a sarcomere or a sarcomere is what? A part of the muscle. So sarcomere, we're talking about just a little microscopic portion of the muscle. And in this sarcomere, we have thick filaments and thin filaments. They're overlapping with each other in a very particular way. We don't see the titin in this picture. I can see if I look really closely, right? See the little Matryoshka dolls, little yellow guys? Those are what? Those little tiny yellow guys are the troponin, and the little white molecule wrapped around the F-actins would be the tropomyosin. So I can see it if I look really, really close. And I know your handouts are useless for this, right? So you'll be pulling these up on your screen and looking at them at home. Now, there's other proteins involved. And let me get to one of these. You hear about it all the time. Jerry Lewis. When I say Jerry Lewis, what comes to mind? Some old guy, maybe, right?
Muscular dystrophy.
Muscular dystrophy, right? So let me talk to you about that just a little bit. There are other proteins involved in regulating your movement of muscle. Yes, the thick and thin filaments have to slide. Absolutely, they have to slide. But there has to be something else, right? If all your muscles did were just slide, how does that give us any power? How does that promote the movement completely of our skeleton? So there are other proteins involved. And look at this with me. This is just a cartoon representation of this. But we're looking at the cell. We're inside the cell. Here's the membrane for the cell. There's the endomysium. Remember, what's the endomysium? It's that layer where? Around each muscle cell, right? And then there are thousands of muscle cells packed within a muscle. Purple is what? Thick filament. And the red is the thin filament. And then there's a linker protein. This red guy right here is dystrophin. Dystrophin. It is a protein that is going to link the sliding filaments inside the muscle to the outside of the muscle. And in individuals with different forms of muscular dystrophy, this protein is deficient. It's either completely deficient or is somehow mutated that it doesn't work appropriately, or is somehow doesn't work completely, so that as a person with muscular dystrophy, the classic type, Duchenne's muscular dystrophy, the most devastating form that we hear about the most, those, and mostly boys, those kids, their sliding filaments are fine. Their actin and myosin is fine. What is missing, what is defective, is this red noodle, this protein, this dystrophin. So the energy of their muscles is not able to be translated out, right? And so no force can be generated, and their muscles eventually are not useful to them. So we see there's a linker protein that also carries this information out of the cell. So dystrophin would be one of the accessory proteins, and there are many of them. That's the one I'm going to mention to you by name. Here, the next slide tells you a little bit more about it. So dystrophin is the protein that is going to transfer the forces as your muscles are contracting. What is it that's going to allow that energy to be transferred out from the sliding filaments to the actual muscle? And there's all sorts of muscular dystrophy. I think one of the case studies that some of you have a choice of doing is on muscular dystrophy. And you may have heard of Duchenne's, the most devastating. There's also Becker's dystrophy, which is a little bit of a less devastating form, and those young men live much longer because it's not quite as disabling. Questions so far? Questions so far? Yes? We absolutely will be talking about troponin. We're not going to go away from this. So we're just kind of still going through the basic pieces and parts, and we'll come back and see how it all goes together. When I say that skeletal muscle, when you recognize that skeletal muscle is striated, what are you seeing? Under the microscope, you see stripes. What's causing those stripes? Yeah, overlapping actins and myosins in those units called sarcomeres. Right? So what we're seeing is overlapping fibers, or overlapping myofibrils. Now, when we look at skeletal muscle, and then later on we look at cardiac muscle, we're going to see that they are arranged very, very similarly. That is, we know that skeletal muscle and cardiac muscle are both striated. So the striations that we see are caused microscopically by the same overlapping fibers. Remember that when we look at smooth muscle later on Thursday, smooth muscle is not striated. So clearly there must be a different microanatomy, a different arrangement of the fibers in smooth muscle. Again, when you see those nice strong striations, right, you know that you're looking at skeletal muscle. Now let's go back and review these letters. I have to tell you, some of these letters I can kind of tell a story with and make sense of. Others of them really, you just have to learn them. You just have to memorize them. It doesn't always make sense to me. So as you look at each sarcomere, let's first define a sarcomere. A sarcomere is from Z to Z, right? So from Z to Z, that is a sarcomere. And then within that little functional unit, again, it shows a stack, doesn't it? I don't want you thinking that it's just a stack of actins and myosins. It's a three-dimensional cylinder of overlapping proteins. And we see the purple are the what? The purple are the thick filaments, and they're arranged in the center of the sarcomere. And then there are, from each side, some overlapping actin molecules. This muscle would be at relaxation. The actins and myosins are slightly overlapped, but they're not completely overlapped. So this muscle is basically at rest in this cartoon. Now, let's go through these bands. I'll start with the easiest one, I think, the Z discs, right? And sometimes you'll see the Z line or the Z disc in different books. But that is the outside border of the sarcomere from Z to Z. And then the M line, medial, middle, easy enough. There's an M line right here, right in the very center of the sarcomere. The A band. Now, the A band basically is wherever the myosins are. So I know it's small in this figure, but the A band is going from here. I'm going right through the A to here. So basically, wherever the thick filaments are, that's where the A band is. The H band is this little area right here in between. Okay? So the H band is in the middle of the A band, and it's where you only see the thick filaments. Do you agree that in the middle, that in this H band, all you have are the purple thick filaments, right? And over in other areas, you have some of the overlapping thin filaments. Another fact is that the Z disc is also doing what? The Z disc is right in the middle of the I band, and the I band is over here, where only the actins are. So the way I remember this, I is a skinny letter, and actin, right, the thin filament. So where there are only the thin filaments, that is the I band. The A band, A is sort of a broad letter, and so that is where only the myosins, where the thick filaments are. There are other names for this, but they don't mean much to us. And the people who have first originally put these terms together were Germans. So I understand that maybe to the Germans in the world, these letters have more meaning. But to us, as English speaking, some of these letters are kind of random. So make up a story as you think about how to remember these different names here. Now, what would you see under the microscope as the muscle contracted? Okay. So if you look under muscle, we see the striations, right? So do you see the stripes now? I mean, there's it. If you were to look under the microscope, you would see these stripes. So that what we're seeing are these overlapping areas of actins and myosins. Let's take a look now. Here is a sarcomere it's marked right here, from here to here. There's a sarcomere. Okay? Let me clean that off. So the sarcomere from here to here. That means that this is what? Z to Z. The A band is where just the myosins are, and because it's a thicker protein, sure enough, it looks denser, right? It looks darker. The M line is going right down the center, and the H band here is very small, but it's that little area just in the center where there's a little bit of a clearing where there are no overlapping actins. And then the I band is out here where you just have the thin filaments. And you've got one sarcomere linked to another sarcomere, linked to another sarcomere. And sarcomeres run the entire length of the muscle. When one sarcomere, remember we talked about all or none idea in the action potentials, right? An action potential is all or none. That all or none phenomenon is also true in skeletal muscle. When one sarcomere shortens, every sarcomere in that muscle will shorten. And what we're going to see in a few minutes is that every muscle cell is innervated by only one axon. Even though these muscle cells are really, really long, there's only going to be one axon, one nervous signal coming into each and every muscle cell. And when that signal comes in, all of the sarcomeres along the entire length of that muscle are going to contract. Okay, so that's going to be bringing that nervous system conversation we've had and bringing it over into muscle. So make sure you can recognize these. Now on Anatomy and Physiology Revealed, there's some help, right? There's some different labeling of these different bands. And if you're struggling with that, go back and look at that.
Actin and myosin myofilaments lie side by side.
Let's watch this.
And the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
Okay, so did the actual myosin get shorter?
No.
No, the myosin, right? So imagine my fingers. Right? If my fingers are actins and myosins, that would make my thumbs what? If this is a sarcomere, my thumbs would be the Z to Z, and my fingers would represent actins and myosins.
Yeah.
So as they slide over each other, my fingers are not getting shorter. So the actins and the myosins themselves are not shortening, but what is shortening is the entire sarcomere through this action of sliding filaments, right? So this has been called the sliding filament theory. It still has the word theory attached to it, right? We know this is how it works, but until muscle was understood, it was a theory that this is how it worked, and it turned out that that is the proper way in which muscle contracts. Would you agree this is about 50% review? And then a few other details to make us think a little bit? So I'm just going to let you read through I've already said everything that's on the next couple of slides. So sarcomeres from Z to Z, right? The muscle shortens as all of these sarcomeres shorten. The thick and thin filaments themselves are not changing length, only that they are overlapping, and that some proteins like dystrophin are going to link that information, pull that power from the muscle and provide it to the rest of the body. So we got the pieces, we got the parts. Now we're going to start talking about how does it work and how does the body regulate this? How do we do this? Well, tropomyosin, can you picture it? Tropomyosin was what? It was that rope-like molecule that was wrapped around the F-actin, and there were two F-actins wound together to make the thin filament. So the tropomyosin and the troponin, I told you, were the regulatory proteins. Tropomyosin is physically going to block myosin from attaching to the actin. So it's going to act as a steric hindrance, as a blocking molecule. And then we're going to see, remember that troponin was like a little Matryoshka doll, little doll in a doll, or a little snowman? There's actually little different subunits in there. So troponin is actually three proteins. There's a troponin I for inhibit, a troponin T, because it binds to tropomyosin, and a troponin C, which binds to the calcium. So that's really convenient, right? The letter tells us what each of those subunits is doing. So here's a cartoon of this tropomyosin and troponin complex. Here, this is probably more realistic. Here, the Matryoshka doll fell over, right? The snowman fell over. So it's really not a stacked three, but you can see these interactions here of these three proteins. Which one is which is not important right now. And remember that each of these globular proteins is called a G-actin. And when they're all linked together, that is an F-actin. Two F-actins bind or twist together and then have along their length this tropomyosin. So that's where we're starting. That's the picture we're going to start with as we think about how this works. I promise you calcium. What does calcium do? Without calcium, your muscles cannot contract. Just like without calcium, you can't have an action potential because there's no release of the neurotransmitter. So when a muscle cell gets stimulated, again, where did it get stimulated? There was an action potential that came down and stimulated the muscle. We'll come back to that interaction later. But as the excitation occurs, calcium will be released inside the muscle. Where's it released from? Where was our storage? Where was our silo of calcium? In the sarcoplasmic reticulum. So calcium will be released from the SR. That calcium will attach to this troponin C, the little subunit that attaches the calcium. Now, when calcium is there, and when it's bound to troponin, it's going to cause a conformational twisting and move the tropomyosin out of the way. It's literally going to move it out of the way. It's going to cause that rope-like tropomyosin to shift. And when it shifts, now the myosin, the thick filament, will be able to bind to and interact with and pull on the thin filament. Okay. Now, in this cartoon, this is the thick filament down here, and this is one of those golf club molecules that are coming off from this big myosin molecule. We see that on the top, there's our actin. We see the double row of F-actins. We see the tropomyosin. When there's no calcium around, okay, when there's no calcium around, look what's happening. This tropomyosin is blocking the myosin from touching or interacting with the actins. Now, when there's calcium, when calcium is around, now imagine that this tropomyosin down here just got shifted. The whole molecule just kind of shifted around. And now the myosin can interact directly with the actin. Okay? As long as there's these little red guys. These are all the calciums. So as long as calcium is bound to this troponin, then that tropomyosin will stay out of the way. Okay, so you tell me what's going to happen. As the muscle is stimulated, calcium will be released. The calcium will bind to the troponin C. The tropomyosin will be shifted out of the way. The myosin and the actin can then do what? Interact, and we'll see in a moment this idea of a power stroke. What happens when there's no calcium? Then the tropomyosin does what? It goes back to its original place, doesn't it? And it blocks the interaction of these molecules. So calcium, 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 very, very important.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges and...
Now, I didn't say ADP or ATP yet, but we did mention how mitochondria were important and in high abundance in cells. So you better believe that ATP, energy, is going to be necessary for this process to occur. And each time a myosin and an actin are going to interact and are going to move, we're going to spend an ATP, right? We're going to have to spend some energy for this to happen.
The phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head.
So what we just saw is that ATP is necessary for what?
For the release.
The disconnect, the
Yeah.
release. That's a very important little tidbit. It's going to be necessary for ATP to disassociate, to disconnect the myosin from the actin. Okay. ATP will be necessary to disconnect. So you saw the ATP molecule fly into the picture, and as it did, it allowed the myosin and the actins to disconnect one from another.
The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
Now you're going to see these videos again, right, in your Connect assignments, and you'll be answering some more questions about them. But a great place to go back and listen to these again. So let's talk about the sarcoplasmic reticulum a little bit more. All right, look at this cartoon representation of a muscle cell. So I know this is the entire muscle cell by looking at some of the clues, because I see the word sarcolemma, right? That outer layer of the entire muscle cell. And within the muscle cell, I see that it's just chock full of myofibrils. And those myofibrils themselves are composed of what? The myofilaments, what you know as actin and myosin. Then we see these triads. Right? What's the triad made up of? It's the sarcoplasmic reticulum. It's wrapped around these myofibrils. And you see this webbing, right? This reticulum. Lots of calcium stored there. So if this is, and then at the ends, right, at the terminus, at the ends of these webs, there is a cisternae, right? A terminal cisternae. And the triad is made up of what? Two of these terminal cisternae with a T-tubule going down the center. So we see that arrangement. Also, look along the sarcolemma. See these little openings? Those little openings are going to be like little rabbit holes. And what we're going to see is that when the muscle gets stimulated, that current is going to basically go along the outside of the muscle. It's going to jump down into one of those little rabbit holes and now interact with the sarcoplasmic reticulum. So guess what an action potential is going to eventually do to a muscle? Fill in the blanks for me. Action potential stimulates muscle. Sodium, we're going to be talking about sodium and potassium in a few minutes. And the sodium, potassium, that potential, that action potential energy is going to go down those rabbit holes and cause the release of what? Calcium. Right? It's going to cause the release of calcium from the SR, and then the calcium is going to do what? Bind to the troponin C. By doing so, it's going to shove what out of the way? Tropomyosin. And now what can bind to what? Myosin can now bind to actin. It will do this thing called a power stroke. It will move and slide the filaments, but it will stay there until another what comes along? ATP. And the ATP will now allow it to disconnect. Some of that energy is being stored. So the myosin will recock back to where it was. As long as there's calcium around, it will once again bind to another actin and will once again do the power stroke. And it will keep doing that as long as there is calcium. Now, why is there going to continue to be calcium? What's going to keep the calcium levels high enough to keep the muscle doing its job? I'm backing up. I'm filling in the blanks. You're filling in the blanks. What's coming into the muscle cell telling it to contract? Action potentials. Right? So as long as you voluntarily are sending an action potential down your motor neurons, telling the muscle to contract, you will continue to release calcium. Now, let's back up. You already know the story. What neurotransmitter was released to cause the muscle to contract?
Acetylcholine.
Yay, acetylcholine, because that's the neurotransmitter of choice for all skeletal muscle.
So is muscle fatigue when you lack calcium?
It can be. There's gonna be a couple slides on fatigue toward the end, and fatigue is going to be many things. One of them can be a lack of calcium, a lack of electrolytes. It can be any number of things. In the extreme, it can even be a lack of neurotransmitters.
Like if you're in the gym lifting and your muscles just give out on you, is that because you just ran out of? 'Cause
Could be.
like,
It could be a glycogen store. It could be ATP. It could be. There's so many things. When I discuss fatigue toward the end, you're going to see that it's not one thing. It's multiple things that can come together to create fatigue. Give me a day. Give me a day. Yeah.
So when the potential goes down the T2 wheel, does it stimulate just one sarcomere, and then because of the stimulation of that one sarcomere, everything?
It's an all or none phenomenon. So within the muscle cell, right, this is one cell. This one cell will only be innervated by one axon terminus. Right, there will only be one axon bringing action potentials down to this one muscle cell. And when it becomes excited, when it becomes activated, every single sarcomere along the entire length of this muscle cell will simultaneously contract. Now, I'm getting ahead of myself, but if you go to lift something light versus something heavy, you are going to do what? Voluntarily recruit a number of muscle cells to contract. So the muscles within my massive biceps are rarely going to all be doing their job at one time, right? Some cells will contract while others are resting. Others will rest while others are contracting. They'll sort of take turns. Now, if I'm trying to lift the heaviest thing I've ever lifted, now I would be recruiting every single muscle cell to do its job simultaneously. And as we'll see in a moment, that leads to fatigue, right? So you can't have all of your muscle cells doing their maximum job, because skeletal muscle, although it responds quickly and is very powerful, it's also highly sensitive to fatigue, right? And you saw some of that as you were doing your hand experiments in lab this week. You know that fatigue is just a reaction to being worked repeatedly. Okay, other questions? Good stuff. I'm kind of jumping ahead, but I'm hoping you see where we're going. And we're going to fill in some of these details as we move through some of this conversation. I've already said this, so I'm going to fly through this. So when a muscle fiber is stimulated, that is, there's been an action potential that has come down a motor neuron. Do you agree it's a motor neuron?
Yes.
This is a voluntary motor neuron coming from where? From your spinal cord, right? Somatic motor neuron coming from your spinal cord, going directly down to the muscle that we're dealing with. And when it becomes stimulated, that calcium will release out of the sarcoplasmic reticulum. But let's say you're a piano player and you want to trill, right? Or you want to move muscles really, really quickly, right? For those who aren't musicians, right? For me to do this, what has to be happening? I have to be sending the signal. The calcium has to be released. Let's go back up. I mean, I'm having to send the signal. Acetylcholine is being released, goes across the synaptic vesicle, comes, across the synaptic knob, and then goes to the muscle. Sarcoplasmic reticulum releases calcium. The calcium binds to the troponin C. The tropomyosin moves out of the way. The little muscles do their little stroke. But then I'm also able to clearly wipe the slate clean, right? So I have to be able to get rid of the calcium very, very quickly. I also have to be able to get rid of the acetylcholine very, very quickly. So even though we slow this down into this molasses-like process, right? This stuff is happening in milliseconds. So we have to, the moment that my brain says stop contracting the muscle, I have to have a way of cleaning the slate and getting rid of not only the acetylcholine from the synaptic cleft, but I also have to have a way of getting rid of all that calcium that's swarming around, right, all of these myofibrils. And so there's an active pumping. I can pump the calcium back into the sarcoplasmic reticulum. So there's an active way of getting rid of that calcium. look back just so you get appreciation of the environment here. Where is the calcium being released from? All of these sarcoplasmic reticula. And it's not shown here, but notice that that webbing is surrounding each and every one of these myofibrils. So basically what you're doing is you're bathing the whole cell and all the sarcomeres in a bathing flood of calcium. So we've got to be able to sequester that back up, suck that calcium back up to stop contraction, to allow us to relax, to do it again. Okay, amazing stuff. So I've already been alluding to this. So we have a nerve signal that's going down to a muscle, and a skeletal muscle will not contract unless it was stimulated by a nerve. Now, 99% of the time, we're talking about voluntary movement, right? But are there times where our skeletal muscles move without our instruction? And we call that a reflex, right? So a reflex is an involuntary movement of a skeletal muscle, right? And so we know that it's not 100% of the time where you had control over it, but regardless, it was still a action potential being sent, right, to the muscle, telling it to contract, even if you didn't have, at that moment, control over it. And then there are disease states, right, like any kind of muscle dystrophy or any sort of Parkinson's disease, where certainly individuals are moving their skeletal muscles but without their voluntary control because there's something wrong in the water, right? There's something mixed up in the system. So we'll deal with that a little bit later. Now, what happens to a muscle if you lose connection to the nerve? And the term is it becomes, there's a denervation atrophy. If a person suffers a spinal cord injury and there's no longer an active nerve traveling to the muscle, then the muscle will naturally atrophy. Okay? So without nervous connection, the muscle will atrophy. Now, people who have had spinal cord injuries will still undergo extensive physical therapy. They will have someone manipulate their muscles. They don't have control over the muscles, but they'll still go through a protocol, and the way is basically to keep their muscles as strong as they can be, even though they don't have control over them, right? To minimize the atrophy, then they're going to keep moving those muscles as much as possible. Now, that could come through a denervation. It also could come through some paralysis that we'll talk about. So, motor neurons. You've already agreed that what sends a signal to a muscle is a motor neuron, a somatic motor neuron. And these are going to be neurons that originated in your spinal cord or up in your brain stem. And they're going to go out to your skeletal muscles. Each nerve fiber, again, what's nerve fiber? Each nerve fiber is an axon. And you know that at the end of every axon, there are some axon terminals. So there is every single nerve fiber reaches out to a number of muscle fibers, but each muscle fiber is only controlled by one neuron. Now, this whole thing is called a motor unit. You saw this term in your pre-lab materials for lab. So here is an example of a motor unit. The definition is one nerve fiber and all of the muscle fibers that are innervated by it. That's the definition of a motor unit, right? One neuron and all of the muscle fibers that are innervated by it. Sometimes one nerve, can innervate a lot of muscles. That would be a very large motor unit. There wouldn't be very much control there, right? But if you had one neuron going down to a big muscle, and every time that muscle contracted, it contracted in large force, it'd be a very forceful contraction, but not a lot of fine control. Other times, it can be a smaller motor unit. That is, one neuron goes down and just innervates a few muscle fibers. The little muscles that control your eye are much more fine-tuned than your gastrocnemius or your biceps femoris. So those would be smaller motor units. So what this is showing here, in a very simplistic way, again, overly simplified, but have you have a nerve coming down. Oops, let me get it. Sorry. You have the nerve coming down, originating in the... Where is it originating in the spinal cord? This is the motor neuron. It's originating in the...
Posterior.
In the anterior root, right? In the anterior root. And within that nerve, there are many neurons. And one of them is blue and one of them is red, just for colors. These are fibers, muscle cells, all bundled within the same muscle. And what you'll see is that one neuron will come down and it will interact with, the red one is going to interact with just all of the red fibers. Right? So each of these are the same size motor unit. It looks like one neuron is going to 4, right? 4 or so muscle cells. But that's a motor unit. So that means that when that neuron sends its signal down, let's say it's the red one, when the red neuron sends its signal down, what will contract? The 4 red muscle fibers. And every sarcomere within that would also contract simultaneously. Okay? Now, how long that signal is sent will dictate how long it will continue to contract. Yes?
I guess I'm confusing the significance of the color differences.
Okay, so red, okay, let me clean it so you can see it better. So what we're saying is that this could be one nerve, right? This is one nerve, right? One spinal nerve. And you know that a nerve is many, many axons. And within this very simple example, there are 2 neurons. There's a red one and a blue one. The blue one is connected to just the blue muscle cells. So if the blue neuron was activated and it was going down to the muscle, only the blue fibers, the blue cells within that muscle would be contracting. 2 neurons, right? Going to a discrete number, a limited number of cells. The average motor unit, 200. Okay, so 200, I mean that showed 4, right? That picture showed 4. But the average motor unit is going to be 200 muscle fibers for every neuron. Now there are smaller motor units. These are going to give a finer degree of control. You might only have 3 or 6 muscle fibers, much like that cartoon, right? A very limited number. Where do you have the most dexterity? Where do you have the most control? What muscles in your body are the most sensitive, most, what's the word I'm looking for? Yeah, fingers, finest amount of control, and like I said earlier, the eye. Those little muscles that control the eye movement, very, very fine, very, very small motor units. Versus large motor units, which would be your large muscles of the leg, for example. Your gastrocnemius is 1,000. Right? So you're going to send a signal to your gastrocnemius to contract. It's going to cause at least 1,000 fibers to contract. It's a lot. There's a lot of force. Not very fine motor, but gross motor. Big motor stuff. Now, within a muscle, there are many motor units. So don't think, here I'm saying there are 1,000 muscle fibers connected to one neuron, but there could be even hundreds or thousands of these motor units within a single muscle. So again, we get a lot of control here. How does it happen? This story is going to look awfully familiar. I mean, this is going to look really, 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 really familiar. If you didn't learn about action potentials before, here's your chance. If you learned it well before, you're going to kick back and say, I've been here, I've done most of this, there's a few things I need to look at, but I got this story down. So, when we talked about the nervous system, we talked about one neuron communicating with another neuron. We call that a synapse, right? Where 2 neurons talk to each other. We talked about that with the sympathetic and parasympathetic nervous system, where there was a pre and a post-sympathetic neuron. We just talked about the overall nervous system. I said one neuron speaks to another neuron. Think about your special senses. There was the primary, the secondary, or the first order and second order neurons, third order neurons. So we know neurons speak to neurons. But we also know that neurons speak to muscles. So the junction between the neuron and the muscle is called the NMJ, the neuromuscular junction. Same idea, right? You've got a nerve fiber, an axon that comes down, little synaptic knob, little axon terminals, and there's going to be a synapse, but not to another neuron, but instead to a muscle fiber. One nerve fiber, one axon, stimulates the muscle fiber at several points within the NMJ, and I'll show you that in a second. Same old story. At the end of the axon terminus, there's a little synaptic knob, a little swollen end, and there'll be little synaptic vesicles all filled with our favorite neurotransmitter, ACH, acetylcholine. It's the only neurotransmitter we're going to be discussing when it comes to skeletal muscle. A little space in between, the synaptic cleft. Schwann cells, we're in the periphery, right? So we've got little Schwann cells that are helping to protect all this. And we'll see that even Schwann cells come around and sort of envelop or protect not only the axon, but they go down all the way down and sort of protect the neuromuscular junction as well. Those synaptic vesicles are going to release acetylcholine through exocytosis. Remember that exocytosis of neurotransmitters also requires calcium. A different collection of calcium, right? But calcium nonetheless is necessary for the release of the neurotransmitters. I know our cartoons always show 1 or 2 molecules. A really fancy one might show 50 molecules, but in reality, there are 50 million acetylcholine receptors on the membrane of each muscle cell. 50 million receptors for those acetylcholine molecules. Now, how are you going to get that many molecules squeezed onto the membrane of a muscle cell? You know the story. When you need more things squeezed into place, you're going to have an increase in surface area. There we go, right? So what we're going to see is that the connection at the neuromuscular junction, the muscle cell is going to have a lot of pleating or a lot of increase in its surface area so that we can squeeze in these millions of acetylcholine receptors. What if you didn't have any of these receptors? If you didn't have these receptors, that muscle can't contract. So if you were to somehow not have acetylcholine receptors or have the acetylcholine receptors blocked by some toxin, then you would have paralysis. You would not, the muscle would not contract. And we'll see some examples of a medication or drugs, I guess, toxins that can interfere with this. And if you block this acetylcholine receptor, then the muscle becomes flaccid, it cannot contract, and you can die, right? You can die from not being able to breathe. If your diaphragm can't work, then sayonara. I mentioned to you earlier that the calcium can be wiped clean quickly because the sarcoplasmic reticulum has a pump where I can suck up all that calcium back up into the SR. Likewise, in the synaptic cleft, there's a mechanism to get rid of the acetylcholine. And I think, Josh, you mentioned the other day when we were talking about this. Acetylcholine can be quickly broken down, partially by an uptake, but also by a breaking down by an enzyme called acetylcholine esterase. Right, so it's an ACE, it's an enzyme, it breaks down acetylcholine, and it can very quickly get rid of that acetylcholine and allow for muscles to go back to a state of relaxation. Again, if you interrupted acetylcholine esterase, what would happen?
Muscles couldn't relax.
Muscles couldn't relax, right? So if you blocked acetylcholine receptors, muscles would never have the chance to contract. If instead you interfered with acetylcholine esterase, then you would never be able to wipe the acetylcholine away, and now muscles would stay in a state of tetanus or in a state of contraction much longer than you would want them to. So we're seeing some control here. We're imagining where could we interact or interrupt this system.
Is that a seizure then? Like if something like that would go on because there's a constant...
Kind of. A seizure would be an uncontrolled release of electrical signal right from a part of the brain that's causing muscles to stay in a state of contraction. It's not because of acetylcholine esterase, and it's not because of a problem with acetylcholine receptors. But that would be an overstimulation of the muscle, wouldn't it? So a person is sending electrical signals unknowingly, right, through an epileptic kind of moment. Those electrical signals are being sent, and the muscles have no choice but to react to that stimulation.
Would it be an example of a Charlie horse?
Charlie horse is more of a cramping, and oftentimes that's more of an electrolyte imbalance. Okay, again, this is all gonna make sense to you about the potassium and the sodium and why you eat a banana soon enough, okay, when you're having a cramp.
Is there an example for this?
For,
Acetylcholine or the
oh, yes, yes, give me a moment, right? Give me a moment. Let's get to the good stuff. Now, let's take a look at the microscopic picture of a neuromuscular junction, right? See ya. So a neuromuscular junction. It's basically the same story as a neuron to neuron, but here we know the players here a little bit better. So what is this? This is a Schwann cell, right? It's coming down, all the way down. There's my axon terminus. Also notice that there's Schwann cells that sort of come down all the way to the knob. That's a little bit different, isn't it? When we discuss neuron to neuron connections, there were never Schwann cells down at the very, very end. But in a neuromuscular junction, sure enough, those Schwann cells are down there, and they provide some extra protection to this area. Okay, no surprising. Some mitochondria, need some energy. These little vesicles, full of acetylcholine. As the action potential comes down, you know what's going to happen. Doesn't even show it here, but what's going to come in?
calcium.
Some calcium's going to come in, right, and cause the exocytosis of these vesicles that are going to release the acetylcholine. A little space, a little cleft in between. Oh boy, the artist got really fancy. There might be 100 receptors there, right? There should be 50 million, right? But we see a large number of these acetylcholine receptors over here in blue. Each of those is acetylcholine receptor. How do we squeeze a lot of them into a limited space? Create a lot of increase in surface area by having this sort of peaks and valley. Now the muscle, this is the muscle cell, right? So this is muscle cell. And again, what do we see in here? A lot of mitochondria. And there's one sarcomere just waiting, right, to be activated by this signal. And we'll break this down, but you tell me what's going to happen. Tell me the steps. Talk to me again. So action potential comes down. What's released? Acetylcholine. What had to come in to make the acetylcholine release? Calcium. Now, it's going to go across, and it's going to activate the acetylcholine receptor. It's going to be received. Now, you tell me what's going to happen in the muscle cell. What do you think? That is going to be a ligand-gated channel, meaning what? When acetylcholine binds to the receptor, it's going to open, and guess what's going to flood into the muscle cell? What always floods in and causes depolarization? Sodium. Oh, we can't forget the story, right? So when I'm getting ahead of my story, but you already know the parts. You already know the story. So as acetylcholine binds to the receptor, that is going to be a ligand-gated channel. Ligand-gated sodium channel. And guess what? Sodium's going to rush into the muscle cell and cause the muscle to become depolarized. That signal is going to travel much like an action potential, and that signal, I'm getting ahead of myself, but that signal is going to do what? Run along the outside of the muscle cell, and eventually, remember I said go down the rabbit trail, down the little rabbit hole? That little signal is going to go down that little rabbit hole and cause the release of calcium from the sarcoplasmic reticulum, and from there we know the story a little bit better. Okay, so that's where we're going. This is not that hard. We just got to pull what we know from the nervous system and bring it into muscle language and tell the story again. Isn't it cool?
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
So that's how we get some of that recycling, right? Get rid of some of the extra, the acetylcholine esterase, and then the neuron can actually put that back together. The story makes sense?
Yes.
Right? I mean, all of a sudden you realize why on day one we talked about sodium. And I said, you'd be sick of it, and you're probably sick of it. We're going to keep talking about sodium and calcium and potassium, and it doesn't go away. So what is this picture? What we're looking at here is a light micrograph of, it says motor fibers. So this would be the motor nerve fiber, right? The axon coming down. And this would be the swelling of the synaptic knob. And it's making interaction with the muscle cell. Remember the muscle fibers? We see them. There's a muscle fiber here. Here's another one here. Long cells with striations in them. We know those striations now are the overlapping actin and myosins that we can see not macroscopically, but we can see under the microscope. Here is a transmission electron micrograph of a neuromuscular junction, and it's a little bit hard to see here, but we always show these big gaping spaces, right, if you will, between the neuron and the muscle. These are very small spaces, right? The synapse is where things come together in very, very close proximity. Okay, so what happens? Muscle is electrically excitable. That means it must have a resting membrane potential. That means, right, what we already know about neurons, that muscle is excitable, that there must be a voltage change across the membrane from the inside and the outside of a muscle cell. And the study of this is electrophysiology. So there are people who spend their entire life looking at the electrophysiological changes of muscle when they're stimulated by different stimuli. Now, what was the resting membrane potential of a neuron? Negative 70. In a skeletal muscle cell, new number to remember, it's not that big a deal though, negative 90. So what does that tell us? That the inside of a muscle cell is more negative than the outside. The difference, the polarity is 90. And the same ions that contribute to this polarity are the same ones we found in the neuron. So again, sodium on the outside, potassium on the inside. That RMP is maintained by the same sodium-potassium pump that we learned about in the neuron. So what happens in an unstimulated or resting muscle cell? This is the same story, guys. Are you bored yet? That's why they're all leaving. They already know the story. They're like, I'm leaving. There they go. They just keep leaving. See you, Jordan. No. So it's the same story, right? It's the same story. You know this. So there's more sodium where? Extracellular. There's more potassium inside. There's also inside more negatively charged proteins and nucleic acids, which make or keep the inside of the cell more negative, and those molecules can't leave. What can leave the cell are pretty much the sodium and the potassiums and the chlorines and some of these smaller ions, and they always move through channels. Okay, so as we stimulate a muscle fiber, it's just like stimulating a cell, a nerve cell, or a neuron. So again, yawn, right? Ion gates are opened. In this case, what kind of ion gate was it again? A ligand-gated or ligand-gated channel. It was a ligand-gated sodium channel. What was the ligand? The ligand's a fancy word for the molecule that when it touches it, it opens. The acetylcholine, right? So acetylcholine binds the receptor. As a result, the gates open, sodium comes flooding in. We know what happens. There's a depolarizing event. Same story. Immediately the sodium gates close, the potassium gates open a little bit more slowly. Potassium now rushes out of the cell. There's a flipping of the electrical charge. We then will repolarize. Again, we know this story. The action potential that he mentioned was on the muscle cell is the same looking thing. It's a quick up and down, depolarizing and repolarizing event. And we'll go back to a resting membrane potential. And yes, there's also a little bit of a similar type of refractory period here as well. What can interfere with this? Yes, sir.
Does it still have to reach to positive 35 before it comes back down? Are the numbers basically?
Let me give you some numbers in a minute, but I appreciate you going there. So just know right now, the only number we've changed is that the RMP is negative 90, right? Okay. Now, we've already kind of alluded to this, and I think you've already thought through it. There are some molecules that could muck this system up. And these molecules, some of them are pesticides. So you hear about people, who work with pesticides who die from some sort of muscle problem. So some pesticides contain cholinesterase inhibitors. So what would they do? It tells us they're going to bind to the acetylcholinesterase. They're going to prevent it from degrading the acetylcholine. So what does that mean? The acetylcholine is going to hang around too long. As a result, the muscle is going to continue to be depolarized and continue to contract. This can lead to a spastic paralysis. So the muscles are in a continual state of contraction, and you can suffocate from not moving your muscles or from not being able to stop them from contracting, right? Either one can lead to suffocation. There's also tetanus, another toxin. This toxin, though, is produced by bacteria, a bacterium called Clostridium tetani, and it creates a toxin which basically blocks glycine release and overstimulates muscle. So tetanus, what is tetanus? Tight, right? You get a tetanus shot to prevent getting lockjaw, from preventing getting this tetanus bacterium or this Clostridium. So we hear about don't step on a rusty nail, right? So where do we get this? Not necessarily totally true, but this Clostridium is a bacterium that is in the soil. And so if you get something that's old or in the soil up into your body, into your foot, then there's a possibility that the Clostridium, not even the bacterium, but more the toxin, can enter your body. And that toxin can then interfere with and cause a tightness, a tetanus, a lockjaw of your muscles. And that too can be deadly. Ever watch any sort of National Geographic specials and you look at the South American Indians and they would have the little blow darts, right? And they were always playing practice with the missionaries coming in. Okay? So the curare is the molecule here. And they didn't know how it works, but they would throw this dart at an animal and the animal would drop, right? It's not dead yet, but it would go into a state of flaccid paralysis. Or if you shoot this stuff into you, and what it does is it competes for this curare, competes for the acetylcholine receptor. Therefore, the muscle can't get activated, can't contract. So the body goes flaccid, right? So if you want to drop an elephant, just give him a little curare and the elephant will just drop, right? So you can hunters use it for wild game, and the natives used it in their blowgun darts in South America. Another beautiful toxin, also formed by a different bacterium, but also has the same genus called Clostridium, botulinum. Now this is a food poisoning. Okay, so botulism, again, you're getting this toxin ingested into your body through your diet. Maybe you're eating grandma's beans, right, from that she canned last year, and something didn't go right during the canning process, and so some of this bacteria got in there, and that's why you never eat a jar, right, that has the top is popped because you've got gases in there which are created by some of these bacteria. So if you ingested this, this botulism toxin will also block the release. Now it does something different, right? But it blocks the release of acetylcholine. Okay, so what's going to happen? If I don't release acetylcholine, then I can't cause my muscles to contract. Okay, so now pharmacological people have come up and now they're sticking Botox into you, right? So they've taken the same botulism toxin and we inject it now in limited areas and it causes your muscles to stop contracting. It releases your muscles. So every 3 months you go in and get a little botulism under the skin and it causes your muscles to become flaccid, to not be as wrinkled. So we're using this stuff now. Dermatologists are getting rich. Okay, let's back up. Any questions so far? You seen the big story? You're not overwhelmed, right? You're really not overwhelmed by this. This is a lot of familiar story with a few extra details added in. Let's take just a couple minutes, refresh ourselves. Let's take about 5 minutes and we'll come back. Okay, so the good news is, ladies and gentlemen, as we come back together, we already know this story, right? We're just kind of piecing together some new ideas. We already know the action potential story. It's just a modification of what we already know. Remember that muscle fibers are going to contract. We've talked about that. And this contraction, we're going to come back to a little bit deeper on the steps of the actual contraction. And there are 4 phases to the contraction of the muscle. First, it gets excited, right? Action potential, it gets excited, sodium channels. And then there's a connection. How do we get from an excitation? How do we get from a sodium potassium to an actual contraction? So we call this the coupling. So there's an excitation contraction coupling where we basically translate or switch from electrical signals to contraction. And then there's the actual contraction, and then finally there's relaxation. So we'll break this down into 4 steps: excitation, contraction coupling, then contraction, and then the relaxation event. I think I've already said this. We probably could go home really soon, but there isn't time for that. So what do we have here? Right? We have to go to lab. That's right. There's no rush. What's the rush? There's an exam tonight. That's where everybody went. I'm a little slow, right? Those folks have lab exams tonight, the ones that kind of are curtailed out here, don't they? It's going to be okay, folks. If you who has a lab exam tonight? It's going to be okay. Who had it this afternoon? And what do you think? Not.
It's not hard.
You know, if you know it, it's easy, right?
Exactly.
Yeah. Okay. So we've already been through this story. So let's recall the story. The nerve signal comes down. Nerve signal, fancy word for an action potential. And we know calcium is going to come in and going to bind. And we know the acetylcholine is going to be released. The acetylcholine is going to bind to the acetylcholine receptors. Those acetylcholine receptors are better called ligand-gated sodium channels. That would be excitation, right? So when the acetylcholine binds, right, the molecule opens up and what comes in? Hold on. There is no calcium here. There was calcium that came in to get the neurotransmitters to exocytose. But here we're talking about the sodium-potassium switch. So as the acetylcholine binds, right, the gate opens, and we know what happens. Yep. Very quickly, sodium comes in, depolarizing event, and I don't like this picture. It shows potassium going out in the same molecule. Works well for cartoon, not good for reality. Okay? But again, sodium comes in, potassium goes out. Now, here's the numbers. The sodium now will shift from a resting membrane potential of -90 all the way up to +75. I still haven't given you threshold, but our curve would be from a -90 up to a +75. Before, our curve went up to as high as +35. It's just the same looking curve, slightly different numbers. Now, we're going to call this an end plate potential. So what is that quick voltage shift? We're not going to call it an action potential, although it is, right? But an action potential is something that travels down a neuron, down an axon. And at the end plate potential, right, action potential, end plate potential is what's going to travel now down along the muscle cell. So we're going to say that we've created a EPP. Oh, I like this so much better, right? So now we see separate sodium and potassium gates, right? And what's going to happen is along, where are we? This is the neuron, and this is the muscle. And we just brought in sodium, and that sodium is going to change the what? There's one, right? Not two pluses, one plus. That sodium is going to do what? Cause a voltage change, isn't it? Now that voltage change is now going to activate additional voltage-gated channels. So same old story, right? That's what we had. Voltage-gated channels were the kind of channels down along the neuron that propagated the action potential. So as that signal now comes along the sarcolemma, it is going to a localized area, cause more voltage-gated channels to open up and bring sodium in, and likewise potassium will go out. And this is going to create a slight negative positive change, isn't it, along the membrane. And just like an action potential propagates, so too this disturbance will propagate along the sarcolemma. This is all still the excitation.
An action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the...
So those T-tubules are what? Remember, they open up to the outside. So the rabbit holes I was talking about, right? Those rabbit holes are the T-tubules, and that signal, now he just called it an action potential. It's okay, but you'll also hear it called the end plate potential. So that is what's traveling along the sarcolemma.
Ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open.
What kind of channels? Voltage-gated calcium channels. What does that mean? That as the voltage change comes down the T-tubule, that voltage difference is going to knock open some gates. Those gates are going to allow calcium to be released from the sarcoplasmic reticulum and now flood the myofibrils. That calcium we know will bind to the troponin C.
In an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament. Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin-binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction.
Got it. All right, we got the story, right? We can tell this story. So that's excitation, right? Getting the signal from the neuron and traveling along the membrane down the T-tubule, right? We've just excited the membrane. We've excited all the calcium, everything's being released. Now we have to couple it, translate that signal, that calcium and electrical signal, into something that's going to actually cause contraction. So as the action potential travels down the T-tubule, right, and there's the T-tubule, comes down, there are voltage-gated calcium channels in the cisternae, and that's going to release the calcium, and now the calcium can bathe, right, the myofibrils and the thin filament with the troponin C, and get everything to begin to contract. So that's what's going on. As the calcium binds the troponin, and as the tropomyosin shifts its location, that's the coupling. Right, that's the coupling activity, and now we'll see contraction begin as myosin can bind to the actin. So contraction will be the actual binding, right? So the calcium came in, bound troponin C, that shifted the tropomyosin out of the way, and now the actin can bind to the myosin can bind to the actin. Okay. Now, does binding require ATP? The binding of myosin to actin does not require ATP. What does require ATP is for it to dissociate, right? So the binding is energy free, if you will. And we're going to be going through this little cocking or this little power stroke story as we go through this. So what happens is that the myosin, when the myosin head releases ADP, so there's still an ADP. Now what's ADP? There's ATP, which is triphosphate, 3 phosphates. And when ATP is broken down, you get ADP plus the phosphate, right? It's called an inorganic phosphate, so you'll see P sub I. So that's ADP. You've taken the energy from the ATP. And so we'll see that when myosin binds to the actin, the ADP will be released, and that is going to cause the myosin to do its power stroke, its movement. Now, once another ATP comes in, that can now cause the myosin to release, and we go back to where it was, grab another actin, but only as long as what? I can only grab another actin if there's calcium present and if that tropomyosin has been shoved out of the way. Each stroke of each myosin requires another ATP molecule. Again, we draw this in one little molecule at a time, but there are millions of myosins grabbing millions of actins, which are simultaneously doing this power stroke deal, right, to give you strength within your muscles.
Myofilaments are composed of strings of the proteins actin and myosin. An actin filament has 2 strands of actin molecules wrapped together. A myosin filament has many myosin proteins packed together, and each myosin protein has a globular head region that protrudes from the filament. Myofilaments can contract or shorten due to interactions between the myosin heads and the actin filaments. Contraction begins with the head of myosin molecules bound to actin on the actin filament. While still bound to actin, the myosin head flexes, pulling the actin filament along with it. This causes the actin filament to slide by the myosin filament. Then the myosin head releases from the actin and unflexes, a change that is powered by ATP. This frees the myosin head to bind with a different actin molecule farther up the actin filament. The entire process may then be repeated many times. The myosin, in effect, walks along the actin filament, moving the actin filament more each time.
Now there it looks like there's specific myosins that are in blue. That's just a cartoon thing. But it would just keep moving along the way. Now I don't know that you'll be able to hear these videos on YouTube. Somewhere, I think I read that it won't work. If it does, great. But somewhere I read that this may not work. So I'm trying it today. And if it doesn't work, then you'll just see the darn picture for 10 seconds. And you can always go back and replay these videos if it helps you. But I'm not sure they're going to play on the YouTube video. There's some issue there. So then we've done the excitation. We've come down and we've done the binding of calcium to troponin and tropomyosin moving. That's the coupling. We've just done the actual power stroke, the actual contraction. And now the fourth and final step is relaxation. So 2 things have to happen. Both the nerve signal must stop, and we must get rid of any acetylcholine related to that excitation. And of course, we've got to get rid of all the acetylcholine from the acetylcholine esterase. And it doesn't say it here, but we've also got to sequester all that calcium, right? We've got to suck up all that calcium into the sarcoplasmic reticulum as well. And that then allows the muscle to relax. And here's the second half. So not only do we have to get rid of the acetylcholine, but we have to get rid of all that calcium. Now look at this. The uptake of calcium back into the SR is active transport, which means what? Even more ATP is used, right? Even more energy is needed to bring your muscles to a relaxation phase. So do you fatigue more quickly when you're doing something quickly? I mean, will my finger fatigue?
Yep.
Trilling or moving something quickly? Yeah, it will tend, because there's a lot of energy not only in the contraction, but also there's a fair amount of energy being used up just to get the darn thing to relax, to keep contracting. So you're actually, creating a lot of need for energy as you do that. So what you see here is that ATP is necessary not only for muscle contraction, but also for muscle relaxation. Let's look at this term. The protein that binds up the calcium is called calsequestrin. Kind of a cool name. What other words kind of built into that? Cal for calcium. It ends in IN. And what is the calcium? What's happening to the calcium? It's being sequestered, right? So a jury is sequestered, right? It's isolated. It's pulled away from society during a trial. So you're sequestering it. You're pulling that calcium away from the rest of the environment. Okay, so once we get rid of the calcium, what happens? The calcium is no longer available to bind to the troponin C. The troponin C then allows the tropomyosin to go back to its original resting position, and that would then block elastin from binding to the myosin.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
If we hear it enough times, we'll believe it, right?
Yeah.
Okay, so over and over and over. If you need to hear these again, just go back and listen to them on Connect. Okay, so that's the physiology. That's the muscle physiology. That's the calcium. That's the sodium. That's the potassium. That's all that's going on at the microscopic level to get your muscles to contract. Now let's talk about some other relationships and how the muscles do it. There is a relationship between the length of your muscles and the tension that your muscles can create, or the power they can create, and what's called the tone of your muscles. If I say to you, wow, you are, boy, I get in trouble for saying this, you really are well-toned, right? Or you notice that someone is well-toned. What does that mean in the everyday language, right? That person's well-toned.
Not a lot of fat around it.
Not a lot of fat. Good muscle definition, right? So if someone is well-toned, what does it mean?
Been using their muscles.
That they've been using their muscles, and that their muscles are in a constant state of a little bit of contraction, which gives them this shape, right? This appearance. So a person who is well-toned is using up ATP, aren't they? Right? So the more muscle you have and the more toned you are, the more ATP that you are using, darn it, at rest. So the good-looking people are burning calories just sitting there, looking good, right? And then there's me, right? Not a lot of tone, not a lot of ATP being spent, it's being stored, and we see it, okay? So that's the story, right? And that's just the reality. Okay, so there's a connection then between our muscles and the length of the muscles, how much force they can contract, and how this all goes together. Imagine if your muscles are overly contracted at rest, right? You're keeping your muscles overly contracted. There's not a whole lot of other work you can do, is there? So if your muscles are already at rest, are contracted, there's not a lot more contraction that can occur. So your ability to pick up something heavy would be restricted, right? It would be limited. Now, what's going on at the microscopic level is that your thick filaments have already slid over some of the thin filaments, and there isn't a whole lot further to go, right? When we think about the sarcomere and what's going on with the sliding filaments. Conversely, if at rest your muscles are too stretched, the sliding filaments are pulled apart from each other, then also there's going to be a weaker contraction because you don't have very much crossing over at the beginning. That means that there is an optimum resting place, that your sarcomeres and your actins and your myosins should at rest be at an optimal overlapping relationship with one another. And this is what gives you tone. Your body wants to be able to do the most work that it can when it needs to, right? So your body is going to invest in making sure that your muscles are at the optimal tone, so that when you are asked, when your muscles are asked to do something, I'm kind of speaking teleologically here, but when you ask your muscles to do something, you're going to get the greatest bang for the buck. So your muscles are always at this, quote, optimal distance. And your nervous system is constantly modifying this, right? Now, this is definitely involuntary, right? But you have involuntary fibers that are keeping your muscles at their optimal distance at any one time. And again, we call this your muscle tone. Look at this graph. We can graph this relationship as follows. Here is a sarcomere length. It's in micrometers, right? So we're talking about how far Z is from Z, right? The whole length of the sarcomere. There is an optimum, right? So what we see is that there is a distance a little bit over 2 micrometers where you get the maximum tension or the maximum strength generated from a muscle. If the fibers are if the sarcomere is too short, in other words, right, they're contracted too much at rest, a short little sarcomere. When you ask this muscle to do work, it can't do a whole lot, so there's not much force that's generated. Likewise, if the sarcomere is too stretched and there isn't enough overlapping between the actins and the myosins, then there isn't much to grab, right, to pull and start that tension. So we want our muscles to stay at this optimal resting length, which is between 2 and 2.25 micrometers. So roughly 2, right? Remember 2. 2 is the optimal number or thereabouts for keeping your sarcomeres. Does that make sense? That idea?
Yeah.
Okay. We want our muscles to be at rest at an optimal overlapping configuration. Now, after life is death, right? And after death, what happens? Shortly after death, the body goes into a state of rigor mortis. Now, we now can better understand what's going on. So upon death, there's no more metabolism of your cells, no more oxygen is entering. Without oxygen, you can't make ATP. And at some point, the available ATP in your cells is depleted. We just saw what happens when there's no ATP around. Your muscles can't relax, right? The muscles are not able to disconnect the myosins from the actins. And so there will come a time where, after death, the sarcoplasmic reticulum, right, will be deteriorating. So not only is there a lack of ATP, but also the cells are beginning to undergo autolysis, right? Autolysis, autolysis, right? So the cells will begin to break down. So autolysis begins. The sarcoplasmic reticulum will start to break down, and that will release lots of calcium. Okay, what's that going to do? Calcium is going to do what? Contract to a point, right? So they're going to contract, the muscles are going to contract, but there's not going to be any ATP around. So they're going to contract, but then they're going to get stuck. right? So the body will go into a state of rigor, right? So now after a while, it'll peak around 12 hours and then will diminish over the following hours. So what's going on? Why is it diminishing? Did ATP suddenly become available? No, but instead the cell continues to undergo autolysis, and the muscle fibers themselves are starting to break down, and so they will naturally relax or release from each other. So we go into rigor because massive calcium is released, causing contraction, and then the lack of ATP, they're stuck there, right? We understand that now. We understand why ATP and calcium are both important for that process. In lab, we basically created a myogram, right? We called it an EMG, right? Electromyogram. But it's a myogram or electromyogram. And what are you looking at here? You know that the muscle is at rest, right? The muscle's at rest. And then, right, you squeeze the hand dynamometer and there was a contraction of the muscles and we saw a greater force, and then there was a relaxation phase, and the iWorks basically drew this more as a hump like this, didn't it? But basically we were drawing looking at a contraction phase. And in lab work, they'll oftentimes use a frog. Remember in the Phil's experiment, they used a frog for some of the action potentials. Frogs have been famously used for muscles as well. And they'll take that big old gastrocnemius, right? Frogs are great hoppers. So that gastrocnemius is the biggest, most developed muscle in a frog. And so they'll use it to study muscle twitches and muscle activity. So again, we know that there's a certain amount of energy necessary to get a muscle to begin to contract. Therefore, there's a what? There's a threshold, right? There's a certain threshold, a certain amount of activity necessary to get the muscle to contract. And then there's a threshold, right? The minimum voltage. And a little tiny quick contraction of a muscle would be called a twitch, right? Twitch. You've ever had little eye twitches, right? Little quick little contractions of a muscle. So a twitch is the definition of a quick muscle contraction. There's a latency period. What's a latency period? It's a time of delay between the stimulus and the twitch. It takes time, right, for all this to happen. It takes time for the signal, for the acetylcholine, for the calcium, for all that to happen. Troponin, actin, myosin, 2 milliseconds. Measure that, right? But still, right, we break this down, but in 2 milliseconds, there's a slight delay. You'd never notice it, right? It's instantaneous to us. But there's a 2 millisecond latency period from the time we stimulate to the time we actually contract. And then the contraction phase would be the time when the muscle is actively, when the filaments are actively sliding over each other. And there's a relaxation phase. It's the same thing, right? We talked about what's happening at the microscopic level. Now we're saying what's happening at the macroscopic level with the whole muscle. Again, there's a stimulation, there's a contraction, there's a relaxation. Now a twitch between 7 and 100 milliseconds. So let's put that into common language. 100 milliseconds is the same as a what? 1/10 of a second, right? So that's how long, 1/10 of a second or less is what we're dealing with as far as a twitch, a really quick muscle contraction. So all this can happen, a 2 millisecond latency delay, and then a 7 to 100 millisecond actual twitch before we return back to rest. We know this verbiage as well. We can have a little bit of stimulus such that there's not enough to reach threshold, therefore nothing would happen, right? A sub-threshold stimulus, nothing happens. If we reach threshold, then sure enough, we'll get a twitch. What's happening? Right? Calcium, the whole story. We have a limited number of fibers that are shortening. Now, compare this back to a motor unit. What does a twitch of the gastrocnemius look like compared to the twitch of a little eye muscle? If there's 1,000 fibers connected to each neuron that goes down to the gastrocnemius, then that twitch is going to be a lot more forceful, isn't it? Right? Still as fast, but much more forceful than if I'm just twitching a little, a few little muscle fibers of the little rectus muscles that contain or control the eye. Okay, let me get through this slide and then, actually no, we'll just stop right here. So we'll pick up with this idea of twitch and continue through this conversation.